Hello, in this video we're going to talk about antibody avidity, what that is as opposed to antibody affinity, and then we'll also talk about multivalency, how multivalency plays out in terms of binding, and what it means to have an avidity bonus. We can follow along with the notes as we go. So you may remember that we previously defined affinity of binding. And the affinity of binding specifically describes the strength of an interaction between one binding site and its receptor. For example, between one antibody Fab fragment and one epitope. So it's strength of a one-to-one interaction. And we often quantify that in terms of what we would call KD for just that one species. Okay, so sometimes people will actually write a little superscript here to denote KD affinity, which means just one interaction. Now, what's interesting is that it's very common to have more than one interaction happening again. In fact, even standard antibodies have two interactions happening at once potentially. And so here we need a different way of quantifying, and we call that method avidity. So avidity is a description of the strength of the overall complex that is formed, even if there is more than one interaction happening. So we can write that it's the strength of the overall complex formation or complex that formed. And sometimes people will call this KD avidity just to distinguish it. Okay, and so avidity is made up of three parameters. Take a moment and try to guess, in class I would pause and ask you, what do you think affects the strength of the overall complex that can happen? So take a moment and ask yourself what that might be. All right, and you may be thinking, well, okay, the first one must be the affinity of each individual interaction. So that makes sense. So here we're talking about the antibody for the antigen interaction, and that would be just defined by that single KD. That's the first parameter. The next parameter would be how many of those events are occurring, and we call that valency. How many interactions can happen? We might call those bonds, but they're really non-covalent, right? So how many interactions? And there's actually a third one, which is how those interactions are arranged. And sometimes that's referred to as the structural arrangement of the parts of the complex. And the reason that this matters will become more clear, but you can already begin to imagine that if you have two interactions happening close together, that may be a very different type of interaction than if you had two that were very far apart and couldn't help each other stay bound, okay? So by structural arrangement, we really mean, is it free in solution versus being bound on a surface? Or is it loose so that it's only one set of molecules interacting versus in an aggregate? So dilute versus aggregated could be a way to phrase that. Okay. So those are the three things that can really affect avidity. We've already extensively covered in the prior videos what we mean by affinity. And so today I'm going to focus on valency and structural arrangement and how those play out in terms of avidity. All right, so let's start with valency. All right, and what we really mean here is the number of binding sites per molecule. And you can ask yourself, how many binding sites are there on a typical antibody? Okay, and you may, if you have been following carefully along, know that maybe you should ask what type of antibody are we talking about, because each antibody can actually have multiple binding sites, and it varies by isotype. So IgG, which is the antibody that we most often think about, in which we can draw with this abbreviated Y shape. Of course, it binds at each of its two Fab arms. And so this is a bivalent interaction because it can potentially bind two antigens or two epitopes on the same antigen. Okay, so this is a bivalent molecule. In contrast, the IgM, which is another common antibody form, another isotype, actually is a pentamer shape. And so you have 5 of these heavy light chain complexes, and then they're linked by disulfide bonds in the FC region. And sometimes there's even additional linkages here. It's called a J complex. And so we have this pentameric antibody. How many binding sites can that have? Well, there's 2 on every one of these monomers. And so this is actually a decavalent complex in that it can bind potentially 10 epitopes or 10 sites on an antigen. So a decavalent binding. And there's others in between. For example, there's sometimes people will specifically try to make dimers of antibodies, and then those would be tetravalent, for example. Okay, and valency goes way beyond antibodies, other receptors. It can be bivalent, tetravalent, and so on. For example, the common streptavidin, which we've learned about before, it binds to biotin. It's actually a tetramer, and so it's a tetravalent binding too, and that's part of why it's so strong in binding. That's just one reason why it can be so strong if you have a multi-biotinylated target. Okay, so we have all this with these different valencies, and it's worth noting that antigens can also be polyvalent. There may be more than one epitope on the binding, more than one epitope on the antigen. So if you have this big protein antigen, it may be monoepitopic, where it just has one antigen, and so that's what we would call that, monoepitopic. It has one epitope. Okay, or we may have a protein that has multiple epitopes, either all the same type of epitope or even multiple different types of epitope, and this is quite common on big proteins, and so this would be polyepitopic. And we could even distinguish whether it's polyepitopic in the sense that it has only one type of epitope, or does it have more than one? Okay, and so both the antibodies and the antigens can be polyvalent. And so then the question is, how does this play out? How does this matter in terms of binding? Okay, so why would polyvalency have an effect on avidity, and which direction would you expect it to go? So let's compare the binding of a Fab fragment, which of course is just having one binding site here, okay, versus an IgG, which has 2 binding sites, okay? And I'm drawing these little dots to represent the binding sites. All right, so which one do you think would have higher overall strength of binding to say, a surface that has many copies of the antigen? Okay, so we've got many, many copies of the antigen bound. These are all supposed to be bound on the surface. Which of these species would have a stronger overall interaction with the surface? So pause and kind of think about that. And then if it's really obvious to you which one, think about why. Could you articulate exactly why and like draw a picture about why the interaction is stronger? So go ahead and pause the video. All right, so you may have come to the conclusion that the IgG being able to bind in more places would have the stronger interaction, and that is certainly true. The real question is, can you articulate why? All right, so what we have here is that the Fab fragment can only really bind, even though there's multiple copies of the epitope around, the Fab fragment can only interact with it at one place. And so you get this one binding site in interaction, which means it's constantly coming on and off in an equilibrium state. where it's come unbound. And so once it comes unbound, it might be able to diffuse away, at which point you've lost the interaction. So the Fab fragment, because it can only bind in one place, can easily diffuse away. And in fact, it's easier for it to diffuse away than to come unbound. In contrast, the antibody, once it gets bound at one site, and so now we have this interaction here. Whoops, here's the rest of my antibody. Okay, once it gets bound at one site, then it's going to be in equilibrium with its next state, which is where now it can actually be anchored at both sites to the antigen. And so that, of course, is a much stronger interaction, and it may rock back and forth on that surface where, you know, maybe one side lets go or one side gets released, but then the other side is still anchored. And so in order for this molecule to diffuse away, so it's basically like walking on the surface of the, but staying still, like there's always one foot stuck, you can imagine. And so in order for this to diffuse away, they have to both come unbound at the same time before it can get away, whereas that Fab is just, it's off and it can go away right away. So that's the effect of avidity. So the IgG, so multiple binding, that multivalency directly leads to a stronger interaction because it prevents loss to the diffusion. Okay, that's really the primary reason. Okay, now you may notice here that it matters how far apart those antigens are spaced. If we had spacing that was too big, then the antibody wouldn't be able to grab those two at the same time, right? It could only reach one, and so then you would lose your avidity bonus. And so this is where we can begin to see that the structural arrangement matters. So we've already talked about valency. That was this first part. Now we're going to talk about structural arrangement. Okay, so this is the second part. Okay, and so the first one is it free in solution or on a surface? And then within that, there's what is the spacing on the surface? Okay, but you can imagine, we just talked about this bonus that you get on a surface. Imagine if instead you just have antigens floating around, and then you have antibodies floating around. Do you get the same bonus that you did on the surface where all the antigens were anchored? What do you think? And the answer is no, because whereas on the surface, the antigens aren't moving, or whatever is bound to the surface isn't moving, and therefore it can't diffuse away. In the free solution case, all of the species have the potential to be lost to diffusion, and so it's easier for this thing to fall apart. If we are talking about this in terms of molecular motion, there's actually an entirely orthogonal way of thinking about this, which is in terms of entropy, and you may be able to look at this and see that in this case where things are soluble, there's higher entropy in the system, and therefore it's harder to bring it together because you have to overcome a greater entropy barrier. Okay, so the fact that, but it really comes down to all the species moving versus some of them being anchored. So you get higher avidity in the case where some are anchored. And you could do a concept check here. Did the KD of the antibody binding to its antigen change? No, but the avidity changed. And so this would be that difference between KD that's for affinity and KD that's for avidity. Okay, we mentioned earlier that spacing on a surface matters. And so that's another way that structural arrangement can matter. And so you can ask yourself, you know, let's say you had some antigens that were sort of far apart versus some that were very close together. versus maybe some I'll draw over here that are really, really far apart, okay? And so let's say that the antibody is spaced like this. You know, in which of these cases do you get an avidity bonus? Not here on the leftmost one. I don't know if you can see my mouth, but we can't get that avidity bonus because there's, in this case, the arms of your multivalent binder, your antibody cannot reach the two species. And so now it's as if it's only able to bind to one at a time. Whereas for these two, you could potentially get that avidity bonus. Okay. So spacing matters too, and it's worth noting the typical distance on an antibody between the two Fab binding arms is about 8 nanometers. And so that becomes a good benchmark for how far apart your species should be that you're trying to bind to, okay? And for those who are interested, George Whitesides published long ago in the mid-90s an excellent text on multivalence in ligand design. All right, and I've pasted the reference for you there in case you're interested. The third way that spatial structure can play out with avidity is that avidity can really cause a stable aggregation. So aggregates are greatly stabilized by avidity. And what we mean here is that if there are more than one place or set of places for the species to bind, for example, if your antigen has more than one epitope on it, just as one example, you know, let's say it's tetrameric, and it's homotetrameric, so this is 4 copies of the same epitope on your protein antigen, and then you come in with IgG, and you know, if there's a lot of these around, let's say we have 3 of them just to say, right? So this now could be bridged by a single antibody, and that could happen all over the place. So the antibodies, because they can bind more than one, now you have all these antigens getting stuck together, and we might get some in here, you know, kind of running behind these other ones. And you could see how this could really quickly become quite a complex aggregate, and it would be very stable because every one of those individual interactions is doing that walk where you know, both arms would have to come off simultaneously, and in this case they would all have to come off simultaneously for this thing to ever disaggregate, and that's just extremely unlikely to happen. And so these aggregates become very stable and difficult to break up. Now, in terms of immunoassay design, that could be good or bad. If you've designed your assay to have an aggregate, we sometimes take advantage of this. And we, in an assay called immunoprecipitation, where you add an antibody, and if the antigen is present, you actually look for huge aggregates to fall out. Oops, immunoprecipitation. I think I have too many I's and T's there, but you get the idea. So, so this immunoprecipitation can be good, but if you weren't trying to form aggregates, of course, it could be terrible. So it really depends on the design of the assay. It's worth asking ourselves, when is this most likely to occur, and when is aggregation like this most likely to become stabilized? And we can think about it especially in terms of monoclonal antibodies versus polyclonal antibodies. So if you have monoclonal antibodies, what does that mean in terms of the number of epitopes that they bind? It means that they only recognize one epitope, right? Because you've got a whole clone, and the entire population of antibodies in your monoclonal antibody solution can all only recognize a single epitope. In contrast, the polyclonal antibody reagent would actually be, it's a mixture of many antibodies, and therefore the population recognizes many epitopes, many different epitopes. What this means is that unlike what I've drawn here in this picture where we had to have an antigen that had multiple copies of the same epitope in order to get this stable aggregation, so that's kind of what I just drew right here, a polyclonal antibody would be able to take advantage of antigens that perhaps only have one or very few copies of any one epitope. But of course, there's many potential binding sites on here, many potential epitopes. And now a polyclonal antibody mixture could take advantage of this and have that same avidity effect, because some of them will recognize this pair, while others would recognize this other pair. Maybe I'll draw it as a dotted line to say it's a different clone. I mean, of course, these aren't clones, but a different, an antibody with a different epitope in the polyclonal mixture. And so that's also an antibody. You could draw yours in a different color. And so, you know, you can very quickly build up. I'll draw another one with wiggly lines. This would be a third color antibody. Okay, and so now this is a way that polyclonal antibodies, sometimes we call this a polyclonal bonus, because they are much more likely to be able to make these high avidity aggregates because they can recognize multiple epitopes on the antigen and therefore can easily have this avidity effect. Okay. All right, so the last piece that I want to share with you today is something called the prozone effect, okay? And this is something where we get this sort of interesting effect with avidity where too much antigen might actually oops, not polyclonal, can actually limit binding, okay? And so here, you might imagine, you know, let's say that you were going to do this in an immunoprecipitation assay. Okay, so what you're trying to do is add in some set of antibodies, so a solution of antibodies, and let's just say it's monoclonal. And then you want to add in some antigen. And what you're hoping to be able to see is formation of a bunch of aggregates. Okay, so we want to see, you know, maybe this is a multi-epitopic antigen, and so we want to be able to see that there is this nice aggregation where you've got each antibody binding to, you know, 2 epitopes, and each epitope is bound to 2 antibodies, and so we get an aggregate. Okay, and of course the ratios here could vary, but this is just one example. So that's what we want. That's the desired reaction. And so then the question is, what ratio of antibody to antigen would give you the right interaction? And then is more antigen actually going to give you more precipitation, which is what we're detecting? Okay. And you can kind of think about this for a moment. Maybe you can even draw a plot of how much precipitate are you going to get from this assay as a function of antigen concentration. So I invite you to pause the video and draw a curve that represents that relationship, and then come back. All right, so let's say down at the bottom that there's, you know, hardly any antigen and there's a ton of antibodies, so that would be antibodies in excess. And then here there's maybe 1-to-1, and then over here it's antigen in excess. Okay, so that's kind of our concentration range that we're working in. So when there's hardly any antigen around, we shouldn't see much precipitate, right? So that makes sense. We should see this nice kind of linear thing, and then maybe as they get to be 1-to-1, it sort of levels off. And then the question is, do you continue to see just as much precipitate once there's excess antigen, because that might be what you might expect, that this would look sort of like a binding isotherm. In fact, that isn't what happens. In fact, if you start adding more and more antigen, you actually get less and less precipitation. It comes back down. So you can actually sort of kill your precipitate formation. You can prevent aggregates from forming by adding too much antigen. And try and draw for yourself why you think that might be. You can pause the video and then come back. Okay, so what really is happening in this range over here, this antibody excess range, is that we have so much antigen around compared to the antibodies that each antibody basically can be bridging two separate copies of the antigen and never have to interact with an antigen that is also bound to another antibody, and it becomes statistically unlikely that you're able then to bind up and form this perfect aggregate that we were trying to form up here. Okay, so you can actually overly saturate the antibody binding site so that there's no statistical probability that you can form this big aggregate. Okay, so that's called the prozone effect, where there's sort of this sweet spot in terms of the ratio of antibody to antigen. And whether it's 1-to-1 or a different ratio would depend on the ratios of the number of, or the valency of the antibody versus the valency of the epitope. But the point is that there should always be this like happy middle ground. It's sort of a Goldilocks effect. All right, so that is it for now. Although I'll give you a quick, sorry, one more thing. The very last piece is that we can actually quantify this avidity effect. And it's called we actually call it the avidity bonus. And this was nicely described again in that same Whiteside's reference that I gave earlier. And we've already introduced these two terms, KD affinity over KD avidity. Hold up. Okay, we've already introduced these two terms, KD affinity and KD avidity, which would just be like these binding strengths that relate to either the individual interactions, this 1-to-1 interaction versus the total complex that got formed. And the ratio of these we're going to call, it's sometimes called beta, and that is your bonus. So it's that simple. And so I could give you an example where if I told you that for a particular antibody, let's say, has a KD on the order of 1 nanomolar for a monovalent interaction, okay, then they tested it, let's say, on a surface that has a bunch of copies of this antigen, and they detect an enhancement of 100-fold, okay? All right, so now I can just ask you, what was KD avidity? Okay, and so go ahead and calculate that. Pause the video and calculate that. Okay, and so this is really straightforward. We know that beta equals KD affinity over KD avidity, and so we can just rearrange to isolate the KD avidity. Okay, and so it ends up being 1 nanomolar divided by 100, and so it comes out to 10 picomolar. Now that is a lot lower affinity. You may remember I told you a good antibody is considered to have a KD of around a nanomolar, so 10 picomolar is really, really good. So this avidity bonus can have a huge impact on the system. And the typical enhancements Just for having bivalent IgG, it typically is 20 to 100-fold enhancement just for being bivalent. So if it can bind 2 copies of the epitope instead of just 1, you already get a 2 to 100-fold increase in avidity, okay? And so then it's worth noting, you know, when an assay is conducted and somebody reports the KD for an antibody, you would really have to look closely at their methods to determine, is that the KD for the monovalent interaction, or did they measure it in a way where they would already be having the avidity bonus and this enhancement was already worked in. Okay, so now that is really it for today. Okay, so just briefly, you can look back. Here were the learning goals. We walked all through all of this. So you saw the difference between avidity and affinity. We learned that there's 3 things that guide avidity. We learned what we mean by valency and how that plays out for antibodies and antigens, and how what we mean by multivalency causing perhaps a stronger interaction. And then we talked in detail about how the structural arrangement of the 2 binding partners can make a big difference, whether it's freer on a surface, what the spacing is on any surfaces, and then how aggregation can be stabilized both for monoclonal or polyclonal mixtures. We learned about the prozone effect, and finally we learned about this avidity bonus. Okay, so if you have any questions, feel free to contact me. Otherwise, you can go on to the next video when you're ready, which is going to talk about the kinetics of binding, how fast these processes happen.